Hey, awesome anatomy students. This is the third lecture in the series, and this one is all about muscle contraction and all the steps that goes into muscle contraction. So let's get started. So this first slide is about the properties of skeletal muscle activity. Basically, you'll need to know that skeletal muscles are both irritable and able to be contracted. And so that's what irritability, contractility means. So they're able to receive and respond to a stimulus, and they're also able to shorten when that stimulus is adequate. And we'll talk about that here in a little bit. So we know that in order to contract, a muscle has to be stimulated by a neuron. And the neurons that stimulate skeletal muscles are motor neurons. And we know that motor neurons originate from the spinal cord. So up here we have the spinal cord, and you can see where we have these motor neuron cell bodies originating here in the anterior horn of the spinal cord. So anyway, we call these, for each set of muscle fibers that are innervated, we have something called a motor unit. And a motor unit, you'll need to know this definition, a motor unit is one neuron and all of the muscle fibers that are stimulated by that neuron. Okay? So, for example, here they're showing you two motor units. Here's motor unit 1 and motor unit 2, right? And if you look at motor unit 1, we can follow, and we need to kind of review what this is called here, this long process. This is the axon, right? And that doesn't show up very good, so I better probably use this. So this is the axon, and the nerve impulse is going to travel down the axon and end up at these axonal endings, and you can see kind of these little axonal endings here. They're also known as axonal terminals, and they kind of interface with the sarcolemma of the muscle fiber. So this motor neuron is going to innervate this muscle fiber, right? Motor unit 2 innervates this muscle fiber, and this muscle fiber, right? So that's a motor unit. It's one neuron, one motor neuron, and all of the muscle fibers that it stimulates. Okay? Now we also need to know what this area is on the sarcolemma where this interface occurs. So where the axonal ending and the muscle fiber, where that interface occurs, we call that the motor end plate. That's the motor end plate. And these motor end plates are specialized for receiving the stimulus from the axon. So they're specialized for receiving the information to contract. And they're specialized because they have lots of those receptor proteins on the surface. So they're ready to receive that signal. And so now we're going to talk about what that signal is. So on this slide, make sure you know what a motor unit is and know the axon, the axonal terminals, the axonal endings, and the motor end plate. And now we're going to talk about what's so special about that motor end plate. And so here we have kind of a blown up picture of the motor end plate, right? So here is our axonal ending, or axonal terminal of the motor neuron, right? This whole thing right here. This is our axonal terminal, right? Here is our end plate. And this is the area of the sarcolemma that's specialized to receive chemical signals for contraction. Okay? So how is it specialized? Well, it's specialized, again, because it has all of these receptor proteins on the surface, and they're specialized to receive a special chemical signal that's going to be released by the axonal terminal. And that chemical signal is called a neurotransmitter. It's going to transmit the message from the neuron to the muscle fiber. And so we know that the neurotransmitter in the muscular system is acetylcholine, or ACH, right? That's our neurotransmitter. So we have all of these receptors at this motor end plate for the neurotransmitter. Okay? So let's kind of go over what happens. So the nerve impulse is going to travel, and we can pretend that the the axon comes up here. So the nerve impulse is going to travel down the axon. It's our nerve impulse. It's going to travel down the axon, and as it travels down the axon, do you remember when we talked about the proteins in the cell membrane that are or electronically gated proteins, that they open and close depending on the electrical gradient across the membrane? Well, we have some of those in the plasma membrane of the axon. So we have these gates here, which are really proteins. And so when this nerve impulse travels down the axon, it's going to open these gates. And when it opens the gates, calcium is going to, the axon, the membrane of the axon is going to become permeable to calcium. It's going to let calcium into the neuron. And that calcium rushing into the neuron triggers the movement of these synaptic vesicles. And it triggers their movement down to the bottom, or to the membrane of the axonal terminal. And so they're moving down towards the membrane, and pretty soon they'll fuse with the membrane and release their contents across that space. And that space is called the synapse, or the synaptic cleft. These synaptic vesicles are the vesicles that contain the ACH, the neurotransmitter. And so let's go through this again. So the nerve impulse travels down the axon. That change in electrical environment opens calcium gates. Calcium rushes into the axon, and the calcium triggers the movement of these synaptic vesicles to fuse with the axonal terminal membrane. And when it fuses there, it's going to open up through exocytosis. It releases those molecules of acetylcholine, or ACH, and they travel across the synapse, and they bind to their receptors on the sarcolemma. So they're going to bind to the receptors on the sarcolemma. Okay? So nerve impulse comes down the axon, the calcium gates open, calcium rushes in, the calcium triggers the movement of the synaptic vesicles to fuse with the axonal membrane, through exocytosis, the vesicles release ACH across the synapse, and the ACH binds with the receptor on the sarcolemma. Now, that binding is going to start another series of events that's going to ultimately lead to muscle contraction. So we're going to move on. And so this binding, as soon as those ACH molecules bind with their receptors, that's going to change the permeability of the sarcolemma to sodium. Okay? So let's just review a little bit about the presence of different ions across the membrane. So if this is our sarcolemma, and this is the extracellular fluid, and this is extracellular. Oh, shoot. Let me erase that real quick. That looks terrible. Okay. So this is we'll say this is the ECF out here, extracellular fluid, and this is the intercellular fluid, right? Or this is extracellular, intracellular. So we know that we have a lot more sodium ions out here than inside. So there's lots of sodium out here in the extracellular space, and we can just draw lots of sodium ions, right? We also know that we have some potassium ions, right, on the inside. Okay, and when at resting potential, when when they're not being stimulated, the sarcolemma, or when it's not being stimulated, the sarcolemma is at resting potential, and the voltage across the membrane keeps it stable, right? We have a kind of an equal voltage on the outside and the inside. However, when the ACH binds to its receptors, it changes that voltage because it makes the sarcolemma permeable to sodium. And sodium's going to rush in. So before, we say it was polarized, right? Very stable, it's polarized. Either side of the sarcolemma is polarized. When the sarcolemma becomes permeable to sodium, and sodium rushes in, now we have depolarization of the sarcolemma. And that depolarization is what we call the action potential. Okay? So depolarization is the action potential. And that binding of ACH to its receptors is what triggers the permeability, the change in permeability of the sarcolemma. So sodium's rushing in, and this is happening from that point at the motor end plate. It's happening from there out in all directions, right? Both directions simultaneously. And that's happening across the entire muscle fiber. So it's kind of like a chain reaction. Now, as it travels, as that action potential travels across the sarcolemma, it's also going to travel down into the inner parts of that muscle fiber. And you remember what muscle fibers are full of. They're full of myofibrils, right? And so let's take a look at what happens. So here's our sarcolemma, and let's pretend that our action potential is coming in this way, right? And so it's not only going to travel across the sarcolemma, it's also going to travel down these holes. And these holes, they're not really holes, they're kind of invaginations of the sarcolemma. These are called the T-tubules. These are called T-tubules. And remember when I told you we were going to talk about the sarcoplasmic reticulum last week? Well, the sarcoplasmic reticulum is this looks like netting around the myofibril. So every myofibril has this sarcoplasmic reticulum net around it. And really, the sarcoplasmic reticulum is just kind of a modified version of the endoplasmic reticulum that are in other types of body cells. So the part that you really need to pay attention to, though, is this triad. So we have the T-tubule here, this middle part, right? This is the T-tubule. And then on the outside of the T-tubule is called the terminal cisternae of the sarcolemma. So this is the terminal cisternae of the sarcolemma. That looks really bad. And so why is that important? Well, because just like the sarcolemma is depolarized by that action potential, as the action potential travels down into the T-tubules, it's going to open gates, right? Whoops. It's going to open some gates here. And it's going to release calcium. So we know that the sarcoplasmic reticulum, I think we talked about last week, its main function is to store and release calcium ions. Okay, so as this action potential travels down, it's going to open the calcium gates. Calcium is going to be released. Oops. Wow, I'm not doing very good today. So the calcium is going to be released from here. So we have little calcium ions being released. This is hard for me to write on here. Okay, so calcium is released. Now what happens with that calcium? Well, it turns out that that calcium is our final trigger for muscular contraction, right? So, again, our action potential traveling across the sarcolemma from the end plate, right? And it travels down the T-tubules. It causes the electrochemical gated channels on the terminal cisterna of the sarcoplasmic reticulum to open and to release calcium ions. So where do those calcium ions go? Well, let's look. Oh, this is a slide that I put in here so that you can go through the whole process, all of the steps, and we might come back to that at the end here if we need to. So where does those calcium ions go? Well, do you remember that in order for muscle contraction to occur, we have to have these myosin heads, right, attach to their bonding sites on the actin filament and pull those actin filaments towards the center of the sarcomere. Well, it turns out that the troponin-tropomyosin complex up here, this protein complex, you can see how it is blocking. And it just says protein complex. You'll need to know troponin, tropomyosin. So these myosin heads have to attach to these binding sites, but they're blocked. The only thing that can unblock those myosin binding sites on the actin filament turns out to be calcium. So that's why we need calcium as a final trigger for muscle contraction. And so what happens is that the calcium ions bind to those blocking mechanisms, to the troponin, and move it so that the myosin heads can bind. And the myosin heads then, down here, are going to pull the actin filament towards the center of the sarcomere, and thereby shortening the muscle, or shortening the sarcomere. And imagine this is happening on every sarcomere on a myofibril, and in every myofibril in one muscle fiber, and in all the muscle fibers in one muscle. So quite a bit of shortening there, causing muscle contraction. Okay? And here's kind of what it looks like from this view, right? Remember the sarcomere? Here's our Z-disc. Oops. So here's our Z-disc. I'm sorry. And here's the Z-disc. Here's our sarcomere, right? Once those myosin heads are able to bind, as soon as they bind, they're going to, it's kind of like a trigger. Once they bind, they're going to do the power stroke and pull that myosin or the actin filament towards the center of the sarcomere. And so down here you can see kind of what that looks like with the overlapping. Okay. Once they attach, we call them cross bridges, because they're kind of like bridges from the myosin filament to the actin filament. Okay? So that is the physiology of muscle contraction. There's lots of steps in there. Use that. I'm not going to go back to it, but go ahead and use that slide to go through the different steps that we kind of went through. I've also, on your agenda, placed two very good animations that goes through all of the steps of muscle contraction. And there's also a document that I placed in there that lists all the steps of muscle contraction. And so please use those resources and go through those resources. And for the next lecture, I'm going to review this before we move on. Remember that we have office hours Tuesday and Thursday at 1 o'clock. I'm sorry I wasn't on last Thursday. We had a scholarship committee meeting and I almost forgot about it. So, but this week, Tuesday and Thursday, if you need me before then, text me. My information is on the agenda. So have a great Easter weekend, and I'll see you next week. Bye.